Hey guys, welcome back to another new video by Biology with Zhang Shen. So today we are going to do the paper 4 theory extended February-March 2023, the 0610/42 paper. So this will be the first paper 4 I've ever done. So let's walk through all of the questions in this paper. So this is 80 marks. So the first question, Figure 1.1 is a simplified diagram of the circulatory system in humans. So they give you this kind of a pathway to all of the vessels that link between each organs. So question 1I, state the letter from figure 1.1 that represents the blood vessel that contains blood with the highest concentration of oxygen. So the highest concentration is B. This has to be the pulmonary vein. This is the one that enters into the heart. Okay, why? Because when we breathe immediately, we will get the highest concentration of oxygen and not the arteries. State the letter from figure 1.1 that represents the hepatic portal vein. So in the new syllabus, we are introduced to the hepatic portal vein and hepatic portal vessels. So hepatic means liver, so you have to see which it comes from. So since vein is going back to the heart, it will be C. Okay, and state all of the letters from figure 1.1 that represents arteries. So it's A, E, and D. So A is the aorta and E is the pulmonary artery, and D is the basically the artery that's going towards the organs, even though there's no name. So this 1 mark only for all 3 letters. Okay, explain the importance of the part labeled Y in figure 1.1. So they are asking for part labeled Y. Okay, so you can see that's the label there. Y is the septum. So since this is a 2 marks, you just have to give state what is Y and what is its function. So the septum is Y, helps separates the left and right side of the blood and keeps oxygenated and deoxygenated blood separate. Okay, and question 1V, state the evidence from figure 1.1 that the diagram shows a double circulation system. So what basically it means, what is double circulatory system? So it means that the blood flows through the heart twice in one circuit of the body. It has also a systemic and a pulmonary circuit. So systemic is basically the normal one with all of aorta, vena cava, all of those are the systemic, and the pulmonary is in the lungs, the pulmonary arteries and the pulmonary vein. Okay, question 1B. Figure 1.2 shows the pressure of the blood flowing through different blood vessels and travels around the body. Venules are narrow vessels that connect capillaries to veins. So to show you this graph, is basically pressure. Is to see the change of pressure between different kinds of vessels. So blood vessel acts in figure 1.2 supply blood to skin surface capillaries and have a role in maintaining a constant internal temperature. State the name of the blood vessel that are represented by the letter X in figure 1.2. So we know that the highest blood pressure is the arteries. So the second one is X and third is capillaries. So X will be arterioles. They technically have already given you a clue, is that venules are narrow vessels connecting capillary to vein, and there's one missing here, so it's arterioles. Okay, explain reasons the changes of pressure seen in the arteries in figure 1.2. So they're just basically asking why is there a change in pressure in the arteries. Okay, so there's a change in pressure caused by the muscle contraction of the heart as the pressure increases when the heart pumps. So it's just basically asking you to say why does this happen and how does it cause the pressure to change over time. Okay, this is part 3. Using the information in figure 1.2, explain the structural adaptations of arteries and veins. So 4 marks, just give 2 for arteries, 2 for veins. You get 4 marks. So arteries is that it has a thick muscular fiber wall to withstand stronger pressure. It's a small lumen to maintain blood pressure, and the veins, it has a large wide lumen to provide lesser resistant flow of blood, and then it contains valves to prevent backflow of blood. So I think these the ones that I choose because it's always the most memorable answers. Okay, question 2. A student investigated the digestion of starch. Figure 2.1 shows the apparatus she used. So from here, there's a test tube A and a test tube B, and dialysis tubing is used to represent a cell membrane. The dialysis tubing material allows small molecules to move across it, but not larger molecules. So test tube A and B were set up as shown in figure 2.1 and placed in a water bath at 37 degrees for 30 minutes. And the liquid outside dialysis tubing in test tube A and B was tested with Benedict solution at 0 minutes and after 30 minutes. Test tube A and B, and here are the results. Okay, so using the information in figure 2.1 and table 2.1, explain the reasons for the difference in the result for test tube A and B in table 2.1. So this is 6 marks. So they want to technically ask you to analyze the question that's been given and also the results has been provided. So they say what makes it different. So I would say in test tube A, the Benedict solution remains blue or a negative result because it has no breakdown of starch. You can see that the Benedict solution is blue at 0 minutes, and after 30 minutes it is also blue. It means that there is no breakdown of starch. And in test tube B, the Benedict solution changes to brick red color as there is a presence of maltose, or you can say there's a digestion of starch going on. So you can see it go from blue to red. So if you know your food test, red is the positive result for a Benedict solution. And the process of amylase is broke down into starch or simple sugars and then into maltose. And then because maltose is much smaller in terms of its size, therefore it's able to diffuse from a higher concentration gradient to a lower concentration gradient. So some people will ask me, why is it not active transport? Because since there is maltose technically is a type of sugar, it's kind of like a sucrose like that that's been actively transported in plants. Because it's not in like a regular system and therefore it's in a dialysis tubing, it's small enough to be able to pass through the membrane, so that's why it's being diffused, something like dye like that. Okay, next question. Complete table 2.2 by writing in the names of the missing enzymes, substrates and products. So this is one of the most famous question and it's 3 marks technically. So 4 marks for every row correct, 1 mark. So pepsin, the substrate will be protein and the product will be amino acids. And the enzyme lipase, the substrate is fats and oils, and the products is fatty acids and glycerol. And trypsin is the alkalinity enzyme for breaking down protein. So the substrate is also protein and the products is also amino acids. And enzyme maltase is the substrate is maltose, and when you break down maltose, it gives you glucose. Okay, state the name of the structure that increases the surface area for absorption in the small intestine. So the only thing we know for absorption, it works in here, is villi or the microvilli. Both works the same. Okay, 3A. Figure 3.1 shows part of the classification diagram for plants. For part 1, complete figure 3.1, they ask you to fill in the blanks. So plants can be classified into ferns and flowering plants. Okay, you must know this. And therefore, flowering plants can be classified into monocotyledons and dicotyledons. Okay. Describe 2 identifying features of a monocotyledon. So it has 1 cotyledon, parallel veins. This is usually the most common answers. There are more to it. Okay. Okay, part B. Figure 3.2 shows the life cycle of a fern. The life cycle of fern has some similarities and some differences compared with life cycles of other plants. The letters represent processes that occur during the life cycle. The haploid stages are represented by N and the diploid stages are represented by 2N. So you can see that the spores are N, adult gametophyte is N, and when it's going to gametes, it's also an N, and then to a zygote, it becomes 2N, and when it becomes an adult, it also means 2N. Once I see this, I know it's mitosis and meiosis. Okay. Part 1. The adult sporophyte has 1,200 chromosomes in its body cells. State the number of chromosomes in the body cells of a gametophyte. So you can see imagine that it's 1,200 chromosomes in the sporophyte, and when it's gametophyte, it's N only. So 2N to N is divided by 2. So 1,200 divided by 2, 600. Okay, identify and describe process A in figure 3.2. So they want to ask you what happened in N, that it becomes spores. So once go from 2N to N, it has been cut into half, and the only process that happens to be like this is meiosis. So process A is meiosis, where it undergoes a reduction division, producing genetically different cells. Okay, that's the answer for 3 marks. One for labeling and one for stating what it does, reduction division, and what it produces, genetically different cells. And state the name of process D. Gametes to become zygote, and reproduction, it's fertilization. Okay, figure 3.3 shows part of the 2 flowers from 2 different plants of the same species. So this is technically kind of like self-pollination, cross-pollination maybe. They give you a clue here. So describe what is meant by the term species. So this is in chapter 1. You must know that species is a group of organisms that can reproduce to produce fertile offspring. And annotate figure 3.3 to show the mechanism of cross-pollination by labeling the structure involved and drawing an arrow to show the pathway of the pollen. So you have to label which is the stigma, which is the anthers, and draw on X. Then you have to draw an arrow to show how cross-pollination works in the end. So it goes from one plant to another, from the anther to the stigma of a different flower but the same species. Then draw an X on figure 3.3 to show where fertilization occurs. You can see the X on the second flower, that is on the ovum itself. Okay. Explain the disadvantages of cross-pollination compared with self-pollination. So disadvantages are it has to be reliant on its pollinators, and there is probably a wastage of pollen that can be used for more fertilization to happen, and definitely there will be more energy required to be used. Okay, question 4. Figure 4.1 is a photograph of a tree that has been girdled. Girdling involves removing a complete circle of a bark and phloem from around the tree. So they give you a clue that the phloem is being cut off. And explain why the area above the girdle in figure 4.1 will become swollen. Use the term source and sink in your answers. So the process for this is translocation. They give you a clue that the phloem has been cut. So the glucose is produced from the leaf by photosynthesis, and then the glucose is only converted into sucrose so that it's not too reactive. So the leaves are acting as a source and the roots are sinks. So the phloem helps to transport the sucrose and the amino acids, but however, because it's been girdled, it forms build-up on top of the part where it's been girdled and not bottom, so it cannot be transported down. Part 2, explain why the leaves in girdled trees are still able to receive mineral ions from the roots. They say the phloem is being cut off, but the xylem is not. So the xylem is much deeper inside the trunk, and mineral ions can still be transported in the xylem. Part B, explain why glucose made during photosynthesis is required for the absorption of mineral ions by the roots. So it's technically asking you why glucose is being used for active transport to happen. So when you see mineral ions, you know it's the process of active transport. So glucose is required for plant aerobic respiration to help release energy, not produce energy. Mineral ions are actively transported from a lower to higher concentration gradient. 3 marks. Okay, state the balance symbol equation for photosynthesis. You should be familiar by this in a complete balanced way. 6CO2 plus 6H2O and will become C6H12O6 plus 6O2. 2 marks. Okay, question 5. The eye is a sense organ that's part of the nervous system. Explain why the eye can be described as a sense organ. They basically ask you for definition. So sense organs contains a group of receptor cells that detects to stimuli. You can give some examples, but this answer can already give you 2 marks. Okay, state the 2 parts of the central nervous system, the brain and the spinal cord. Okay. Figure 5.1 is a diagram of a section through the human eye. Okay, draw an X on to identify the blind spot. So it's between the fovea. Okay, the retina and the retinas of both sides. So it's in the middle lah. Okay, and table 5.1 shows some parts labeled in figure 5.1, their names and their function. So they ask you to basically fill in the blanks again. So each row 1 mark, correct. Okay, name of the part. They say the letter is H. H is here, is pointing to the retina, and the functions is containing light sensitive rods and cones. Then the function for this is to transmit impulse to the central nervous system. This will be the optic nerve, which is in G. So that's the answer. And cornea is D. You can see how you remember this is by you can see a C. That's called the cornea. So the cornea, it helps to refract light. Okay, state the name of the 2 effectors that contract and relax during pupil reflex, not accommodation, yeah? So the answer is circular muscles and radial muscles, 1 mark. Explain why a person is unable to focus on distant objects if the suspensory ligaments become permanently overstretched. So if you don't know how to answer this, just answer like why he's unable to focus on distant objects. So how does it happen? The ciliary muscles will relax and the suspensory ligaments can no longer be tightened because you cannot focus on distant objects, only nearer ones, and making the angle of refraction still unchanged. Okay. Okay, question 6. I think this is the last question. Golden rice is a type of rice that has been genetically modified to contain a nutrient called beta-carotene. Figure 6.1 shows the genetic modification process used to produce golden rice, and with a bacterial cell and a plant DNA. So you can see that there's a process of the entire genetic modification that it becomes a tissue culture in the end. So using the information in figure 6.1, complete the sentence to describe how rice is genetically modified to contain beta-carotene. So this is about your understanding on genetic modification. So the part labeled A in the bacterial cell, you can see this is plasmid. So part A is extracted and used and cut using a restriction enzyme, forming sticky ends. The part labeled B is this one, is a gene, okay, that codes for the production of beta-carotene. If we don't know why is it gene, because the definition of gene in inheritance is that it's a length of DNA that codes for a protein. So beta-carotene is for here will be a protein. The enzyme used to cut part A is also used to cut part B from the plant DNA. And then part B is inserted into the part labeled A using the enzyme DNA ligase. This forms the part labeled C, which is a recombinant. Part C is put into a bacterium. The bacterium is taken up by rice plant cells, giving them the ability to produce beta-carotene. So tissue culture is a form of asexual production, not sexual, because you are forming genetically identical cells. And that is used to produce many identical rice plants producing beta-carotene for commercial usage. Okay, that's all. 7 marks. 1 correct, you get 1 mark. For part B, apart from structural features, state 2 reasons why bacteria are useful for genetic modification. Because it's rapidly produced and there's no ethical or welfare issues occurring. Part C, discuss the disadvantages of genetically modifying rice plants to produce beta-carotene. This basically means the disadvantages of GMO crops. So the additional nutrient content may be too small when it's been genetically modified. The seeds to purchase will be expensive, and once you plant too many GMO crops, there will be no biodiversity, and there's a possible risk that the GMO crops can pollinate or cross-contaminate to wild rice or just basically normal rice produce. Okay, last question, part D. Beta carotene is required to produce vitamin A, which is essential for eye function. State the nutrient required to prevent scurvy, which is vitamin C, and rickets is vitamin D or calcium. 2 marks. Okay, so pretty much that's it for this paper. It's honestly not really that difficult for a first February March 2023 paper, but hope you understand. If you do not understand any of these, please comment down in the comment section. I would help to answer and rectify immediately. So I hope to see you guys in the next video. Bye bye.